Are we?
Yes.
Where are we?
Here.
Why are we here? Not entirely clear. We are misfits thrust into existence by random chance, with no hints at all as to how we're supposed to make sense of it all. It's immensely bizarre. Here we are.
Hello, everybody, and welcome to the Here We Are podcast. I'm very excited to talk with my guest today. Denita Hadziabdic Guerry is joining me for the second time because we've recorded once before. And where were you? Your back porch or something like that at home, right? It was.
I was at home, and I promise I've never had those issues before.
And something happened. Well, I was using a different system. And whatever it was between the two of us, oh my gosh, it was, I was going to try to make it work and stitch it together, but it was going to take my editor like 2 full days to correct all of the issues that we had. And so Denita was kind enough to find time to come back on this show to have this wonderful conversation. I'm going to do it in kind of a little bit slightly reverse order. I want to give like a little bit of a spoiler toward the end because I thought one of the really cool, interesting things about our conversation, there was a bunch of cool things that we're going to be talking about, very fascinating research that you do. But one of the really interesting things was we took a simple act of, hey, this is why you shouldn't, if you're out camping and you're going out of state or whatever, you shouldn't take firewood from where you're at. You bought a whole big 3 cords of firewood. You got a big deal on it. And now you want to take that firewood 200 miles away and go camping. And the campground says, hey, use local firewood only. And you might be like, well, what the heck is the difference? And we kind of broke down some of the intricacies of why that's a thing, why that's something that we need to be mindful of. We went into a whole bunch of other stuff as well, but it was fascinating to me that all of it, everything that we talked about was also connected to that, like at the end of the day, the big takeaway punchline for everyone was buy the firewood locally in your local area. So why don't we start off kind of like we did last time and just have you introduce yourself and your research to the good people listening.
So thank you for meeting with me again. You just
what was that? Like
again.
It was like 2 months ago or something, right? So we're in luck because 2 months ago, I'm sure I forgot half of it. So it'll be all fresh and exciting to me all over again.
3rd time's the charm. My name is Denita Hadziabdic Guerry, and I'm a faculty at the University of Tennessee Department of Entomology and Plant Pathology. My research focuses on host pathogen interactions, preservation of biodiversity of native plants, conservation efforts, and population genetics and population biology of different fungi and plants, basically. More recently, I started working a little bit more in microbiome. We have several different projects related to that, and that's really exciting. But generally, what we're trying to do is to preserve native trees across the United States and globally, and prevent diseases from further spread.
So I've been seeing there's a lot that's been going on since we last talked. Maybe it's because I'm now have that kind of confirmation bias, or you learn something new and it kind of changes the way that you perceive the world and certain things get highlighted. And what I've been seeing a lot of since we talked and kind of sort of lost the recording, but talked a lot about some of the different tree diseases and funguses that affect their health and population. And since that time, over the last 2 months, I've actually been seeing a fair amount of stories about wood prices going up, or maybe that's partially a seasonal thing, or is it only because I talk to you and my conscious experience of life is such that now I'm noticing things more like that? Or have you noticed an uptick as well of those reports in the news?
I think it's a combination of things as well. Part of this is pandemic gave us slightly different hobbies. So I'm sure the wood and woodworking isn't on a rise, which could be one of the reasons. Again, I'm just speculating on that part. I'm not 100% sure. But yes, the diseases will do things like that because demand for instance, we do research with walnuts. So there's a disease called Thousand Cankers Disease. And it's a complex disease, which means it involves the host and the vector. And now we have alternative vectors, not just one. We have multiple vectors, insect vectors, that can carry the pathogen, which in this particular case, it's a fungus. And then you have multiple hosts. And in this particular example, also, we have multiple walnut species being susceptible. So walnut is highly prized for its wood and wood quality. It's really heavily used in woodworking, and oftentimes people would invest in walnut orchards as a long-term investment. We're talking 50 and more years, and they use that as a retirement property, actually, because a single good quality walnut tree can be extremely pricey, and they're bred or selected really for straightness of the trunk, for the quality of the wood. So the good quality wood can be expensive. And if people are investing in this over the next 30, 40, 50 years, losing that property is a really big problem. So that is part of the issue we're dealing with. So in this particular disease appeared in the Western US in the late 1990s, early 2000. And then basically in 2010, it appeared in Knoxville, Tennessee. And the eastern border of the Western progression was basically New Mexico. That's as far as it was in New Mexico, Colorado. That was the border of the Western states. And for instance, we know that the pathogen or the beetle cannot fly that far. So this was one of those human-mediated movements. And we talked earlier, I don't know which take was that, about do not move the firewood. And this is a really good example of please do not move the firewood. Because this is how more likely we had multiple introductions multiple times, because both beetle and the pathogen are very genetically diverse, and that indicates more likely that they are introduced to this part of the world, meaning eastern part. That's where I'm at, multiple times on multiple occasions. So that is the interesting part. That's a long answer to your question.
Oh no, the longer the better. It's a fascinating topic because you have in a given area, you have this host, parasite, insect. You have this kind of ecosystem. This maybe not necessarily symbiotic, but there or I think we kind of talked about the Red Queen effect of the kind of arms race between hosts and parasites, and trees in a local area might develop some defenses to certain whatever it might be in the area, beetles or whatever else. But then you go and you move that species into a whole other area where generations of trees haven't had the chance to adapt to that specific threat, and now this parasite or bacteria or insect or whatever that's gotten really good at having to outwit the defenses of a tree in a given area, and now it's in an area where these trees are defenseless. They have no kind of natural defenses. We kind of had a episode about murder hornets last year, kind of a similar-ish situation going on of these Asian giant hornets where the bees in their native area have this amazing defense that they've evolved over many, many, many generations where the giant Asian hornet will come in, and oftentimes if it goes out and gets its buddies soon enough, it can kill all of the bees, but the bees have adapted this defense where they can swarm the giant Asian hornet and kill it before it has a chance to go out and give up the location of where they're at. And sometimes they get it, sometimes they don't, but it's this arms race that goes back and forth. And then all of a sudden the giant Asian hornet comes to the US in a shipping container or something. And and the bees here are completely defenseless. They don't have, they've never seen anything like this. They don't have this mechanism in place to stop the giant Asian hornet from coming over. And then theoretically, this hasn't happened yet. Then this is a great kind of fertile, abundant ground for the giant Asian hornet, and it can explode into this invasive species and really change the ecosystem in a given area. And sort of similar-ish things kind of happen with trees and
absolutely. And it's pretty, like, the whole adaptation as a part of evolution is, I find it extremely fascinating. I think that's where you can nerd out like there is no tomorrow.
For sure.
So similar things happen with, for instance, most people are familiar with dogwoods. Dogwoods are beautiful ornamental trees. They're one of the first bloomers in the eastern United States. They're native here. And there was a pretty devastating disease that wiped off almost all dogwoods. It was called dogwood anthracnose. And actually, that's what I did for my PhD. I did a population genetics of the trees, not the pathogen. And in certain areas, the mortality rate because of this pathogen, which was introduced, actually, pathogen from Asia, was almost like 98% mortality rate. So we were pretty fascinated with that. And you were thinking, this is what I usually use with my students. Sometimes it's easier for them to think in terms of humans and animals because plants do not move, and it's really hard to think about plants and pathogens in that way. And I usually tell them, like, if you have only 2% of individuals staying on the island, for us to repopulate that island is going to take some time. Yeah, it's possible, but it's going to be a lot of inbreeding, more likely. It's going to be a few things happening in between, but it's going to take so many generations to get a healthy, new, viable population. So now you're having the same things with trees. You have 98% mortality rate in certain areas because of the pathogen pressure. You're thinking your genetic diversity of the tree is going to be decimated. You only have 2% of this population alive. Again, in certain areas, this is a native tree. The pathogen is introduced. It does not have any enemies. It does not have any biological control. The Kusa dogwood, which is a variety basically of the Asian origin, that's the dogwood that's originated in Asia, is quite tolerant to this disease. It is not affected as much. It does get it, but it's not as deadly. And they were not as affected as native dogwood, which is Cornus, Florida. So when we did population study, to my surprise, and it took me 4 years to tell you that, so the genetic diversity is not reduced, actually. And we have 4 genetic clusters of dogwood trees, and they're more or less almost like one really big genetic neighborhood. You have 4 different genetic clusters, but there's quite a bit of gene flow within the eastern population of dogwoods. And that's simply, again, evolution is a beautiful thing. You have birds that consume the seed of dogwoods. And when they do migrations, they stay in the digestive system for at least 24 hours. And as they migrate, they deposit that seed along the way and along the migration route. That was at least our hypothesis. And they collect these seeds from random trees along the way. And more likely, those are survivors. Those are pretty strong trees that were not as affected. And this is how you more likely ended up with a high genetic diversity along the way and preservation of biodiversity. And even though you had a high mortality, these trees are going to survive no matter how big disease pressure is. I mean, to me, that's pretty absolutely fascinating that that trees are able to respond that way. Or, trees have certain natural defenses. So, for instance, we're working with oaks now. And oaks have tylosis, which, if you think about that, they're almost like balloons. And when the pathogen enters the host system, then they basically use this as a first line of defense, and nothing can go through, including the nutrients as well. And then this is the first line of defense. Also, another line of defense the trees have that's pretty fascinating. It's called hypersensitive response. So when the pathogen, for instance, lands on a leaf, the plant itself induces the cell death of the surrounding cells, so the pathogen cannot spread any further. I mean, think about that as an adaptation. I think it's so beautiful and amazing. So plants and trees in general, they have a variety of ways to protect themselves against different invaders. And they've done this pretty successfully for millions of years. But every now and then you have a system like that, like what you were using, that just overpowers the host defense systems, or the trees weaken because of the climate change and the lack of nutrients and prolonged drought periods. So the tree's already stressed, and we're seeing that quite a bit. And we're seeing the effect more likely of changing in microbial communities as a result of a drought, including coming back to TCD, Thousand Cankers Disease. We've seen much bigger progression in the West, where you have arid, dry climates versus the East, where we have a little bit different winters. And so all of that goes back to so many, it's very interconnected, everything from pathogen pressures to climate change to drought conditions to host defenses. You have stressors, and trees can handle so much, just like us. We snap at some point when the stressors become overwhelming. So you can fight certain infections, but at some point the body breaks down if it's too much.
Right.
So trees are the same.
So this is kind of the difference between the health of the ones in the East and the West, and in this particular case is that the ones in the West are kind of undergoing this kind of two-pronged pressure, where it's not just the thousand canker that they're having to worry about. They're not getting enough nutrients to help defend them against that, right?
So there are a number of theories and ideas, and it's the whole system. I find it, not just because I work with it, I just find it super fascinating. So number one, it's a complex disease. So I have to work with people that are experts in entomology. Is not my background, for instance. So I have a fantastic collaborator that I work with, who's an entomologist. Then you have to work with, we just started working with a physiologist. Like, that's really not my background. But now we started collaborating and doing some drought experiments potentially, and seeing how drought affects this. But going a little bit larger and bigger picture. So disease appeared on walnuts, all walnut species in the Western US. So walnuts are not native in that area. They are planted. Black walnuts are not native in that area. So they were basically taken with pioneers during the big migrations, and they were planted there as a food crop. So now you're moving a native tree from this area to non-native area. Now you are inducing different host responses. The tree does not have a natural defense mechanisms. It's going to take some time for adaptations to occur to that environment. And what they believe, what scientists think at this point, is also the pathogen that's involved in this complex. It's called Geosmithia morbida. But it's really interesting, talking about evolution and adaptation. So this pathogen was the first pathogen in the species, in the genus Geosmithia, that ended up being pathogenic. That means it can induce disease. And that is pretty interesting, because this group of fungi basically lives in symbiosis, or some version of symbiosis, with other ambrosia and bark beetles. They've never caused disease before. So when did this trigger happen? When did this switch in pathogenicity genes happen with this particular pathogen? And that is pretty fascinating from the genetics standpoint. So something happened and something triggered this pathogenicity. And we actually think that the beetle is native to the Western US, but we think that there is some coevolution between host and the pathogen, because we were not, until actually this year, we were not sure if the pathogen was introduced here or this is the native pathogen. But based on the molecular data, we support the hypothesis that basically this pathogen is native to the US as well, just like beetles, but something triggered that pathogenicity. And that's something that what we're looking at right now, these genes that are involved in pathogenicity and how they trigger basically disease. Why? And we're testing basically a few species that are pathogenic in the same genus and some species that are not pathogenic, and to see how they differ on a larger genomic level.
So you can use molecular data, and now with all of the as technology continues to advance and our understanding continues to advance, you can trace some of the roots and the source of a particular pathogen to see some of its origin, what part of the country that it started in,
yes.
Essentially.
And that is another really cool part of what we do. So that's part of the population genetics. And you can do evolutionary histories and provide a plausible explanation of the origin of your species of interest with certain statistical support. And you can say more likely the pathogen originated from, Western United States, this many hundreds, millions years ago, and so on and so forth. It is pretty fascinating, but also on a bit more simplistic scale, we can use population genetics. And for example, this disease appeared in the late 1990s, early 2000 in the Western US, or at least that's when we noticed the symptoms. It could be that disease was there much longer. And then in 2010 came to Eastern US, so Tennessee was the first state. And then in 2013, it was found in Italy. So you would think Italy was the newest introduction. Maybe this was just a single introduction in Italy. Maybe it was just human-mediated movement, maybe infested wood, because Italians use walnuts for caskets. So, and among other things, but that's really widely used. And you're thinking maybe just one introduction or very few introductions. But when we did population genetics of the beetle, we found that genetic diversity is really high, which you would not expect genetic diversity to be high in newly introduced area. So again, to put it in more simplistic terms for us, if you put two individuals in an island, for them to produce like a larger diverse population, it's going to take some time.
Yeah.
But you only have a few individuals there, so their diversity is going to be limited. But what we found was really high genetic diversity.
Yeah. I was
like, that's.
So if you're just shipping some walnut over to where was Italy?
Yes.
So if you're just shipping a container full of walnut wood for caskets to Italy, and it has some of these beetles in it, and it's just this one-off thing, and that's how it happens, you wouldn't expect such population diversity in that area. So because you're finding such population diversity, you need to get down to the bottom of what the heck happened. This wasn't just a one-off event where something came from the US.
So it's either, so we can have two options here to hypothesize. It's either multiple introduction from multiple sources, meaning from multiple, if it originated in the US.
Yeah.
That is option number one. So we had multiple introductions into Italy. Option number two is
convergent.
Or it's been in Italy for a while undetected. Yeah. Or number three, maybe Italy is the original population. I mean, you cannot exclude that either. So that's what we were working on. And hopefully we'll wrap up that paper sometime soon.
Very cool.
Famous last words.
Yeah. Well, how fast do trees, I mean, going back to humans, and you think about some sort of bacterial threat, or say ticks with Lyme disease, or what have you, you think about kind of that those generations happening really quickly compared to our lifespan and evolving really quickly, and human generation being, what do we consider that, like 30 years or something like that, around the average of when we're combining our DNA and having offspring and
Progeny.
and having progeny. What about with trees? How, because trees have such a long lifespan, but they are also having progeny pretty early on, right? From a pretty early age,
So it
depending
depends
on the
from species to
Yeah.
species, and that's a really hard question for me to answer. I think it's really species dependent what you work with, and how long they're going to take. And considering this with, for instance, insect vector that we're working with, and it depends what region you're in, but you can have 3 to 4 generations a year.
So we have this. I mean, I think some of the stuff that you seem to work on, and some of the. So actually, this is a good time to. Let's talk about the One Health initiative, because I think it will tie into my next question a little bit. Can you explain to the audience? I've had I think you're now my 4th or 5th guest on from the One Health initiative. Can you explain to the audience what the One Health initiative is?
It's a very holistic approach to health in general. So it's not just, it combines everything from environmental, animal, plant, human health in general. So it's all connected, and it's not like it used to be that we're in little silos and animal or vet school is just alone, disconnected from plant health. Plant health is disconnected from soil scientists. Soil scientists are disconnected from entomology people. It's really holistic approach to health in general. So that's how I personally see it. Everybody has slightly different version of explanation of that. But in my case, I work with a tree. Again, we work with a number of things, but I'm going to again use Thousand Cankers Disease as an example. So we work with a tree that's economically important, as well as environmentally and ecologically. So we're talking about socioeconomic importance for people. It provides food source for a number of animals. It is important in the sense of it provides economic stability, but also for overall health, because we're using walnuts, for instance, as a food source as humans, not just for animals. So it's a really big circle of, and then drought and changes in climate change that all affects the food resources and so on and so forth. So it's much larger and bigger than just a tree gets a disease, and it's a complex disease that involves host pathogen bacteria interaction. So it's much more holistic approach to forest health and health in general. So that's my version of One Health.
Great. So I kind of wanted to ask, because I think we've talked about this with some other guests, but one of the not just in terms of climate change, which the Earth has changed temperature in the past and been warmer than this in the past, but this is pretty rapid compared to most of those past times. But in addition to that, this is very much kind of the first time where there was things like global travel and things like some beetle or virus or whatever else could get halfway around the world in a day and all of a sudden be introduced to a non-native population where probably a lot of times when that happens, nothing happen, probably just doesn't do well in a given environment or whatever. But once in a while, there's an invasive species that's really going to thrive. And this is, I mean, there wasn't species carrying things. I suppose there was birds probably carrying some stuff around through migration patterns and stuff, some distance here and there, but nothing quite like what's happening with humans now. And boy, it's definitely concerning to think about. The more, I mean, obviously COVID has a lot of us more mindful of these sorts of issues, but what what do we do exactly? Are there things in terms of what do we do is a little too broad of a question here, so let's be a little more specific. What are some things specifically to say Thousand Cankers that happens that are there things that you go when it's found in trees? Are there things that you can do to help the trees? Are there things that can be done to stop the spread?
So before I answer that, I think you have to answer your question from the little bit broader perspective.
Mm-hmm.
You ask what we can do. I mean, this is a really good lesson for all of us. We had a shutdown of the entire planet for the last year. And I don't think any of us have ever experienced this. This is just what you saw in the movies or read in books. And this is not a real thing, but obviously it is a real thing. So it goes, not to get political about it, but it goes back to importance of science and funding for science. So an importance of educating, starting from elementary school, educating proper science. And getting people interested in science, and especially women and minorities. I'm very, very passionate about that for a number of reasons. And the data supports that women, especially, are by the 6th grade told that they're not good enough, smart enough for science classes, and that's when they start dropping. And that's where we lose girls in STEM discipline. So I would love in my lifetime to see change in that, because now you have more people interested in science, more people interested in pursuing these careers. That means that universities are going to offer more and better classes. That means there will be funding to conduct research for graduate school and advance. We're not going to be a leading scientific force if we continue cutting funding. If we don't go off the wall, cutting edge science and going for things, because what happens with each administration, unfortunately or fortunately, you have to refocus the funding towards whatever the administration wants. Sometimes it's very progressive and sometimes it is not. So in the previous administration, we couldn't use the word climate change in our scientific writing. It was highly discouraged. So you can't shift your scientific research objectives every 4 years.
Are you telling me that the party that was the champions of free speech was scared of the word climate change? The hell you say?
And science-based. Those are 2 words you couldn't use. I mean, that's scary. Mm-hmm. So that should be completely bipartisan, in my opinion. No matter what you believe, no matter how strongly you believe, it should not. So we have to prevent future pandemics. And this could have been easy prevention. We could have had vaccine. We could have done all sorts of things before, but it all goes back to education at the end of the day. So yeah. I'm a very, very strong proponent of education in STEM. And then you have more voters who believe in education and the scientific thought. And no matter where you lean, you have your scientific background. So I think that's the more people we educate, the better we're going to be as a nation in general. So that is kind of a big umbrella approach to that. On the other hand, for Thousand Cankers Disease, so ultimately, when you work with any disease system, ultimately the goal is to obtain some version of control or provide some control measures or prevent or mitigate further disease spread or use that as a learning tool for other similar complex disease systems. So we're trying to do all of that. The complex issue with working with trees is, first of all, you have a number of limiting factors. For instance, we cannot do the field trials and inoculation of pathogens in Tennessee that we found in Colorado, for instance, because this is a federally regulated pathogen and we have certain permits to get the pathogen and work with the pathogen. It has to be under certain conditions. So for us to do the greenhouse trials, which is we have to use only Tennessee strains and we have to have certain preventative measures, which is a fantastic actually idea. So we do not spread this disease any further. So the measures are there for a reason. So that also limits what we can do. And in the greenhouse, we can only work with juvenile trees. But we do not necessarily know if we have juvenile resistance, which is a real thing, or do younger trees versus older trees, maybe they completely respond differently. And you can only test those isolates that you have in your own state, which again is one of the reasons you don't want a reintroduction of or introduction of novel strains and so on and so forth in the field. So research is quite limited in what we can do because of the regulations, which can be a good and bad thing at the same time. So we're trying to do a number of different things in the greenhouse, and we've done some phenomenal studies, but we do not necessarily know if that's going to translate into the field entirely because we work with smaller and younger trees. So also one fascinating thing that we found in the East versus West, we found that trees in the East, when we did culture-dependent method, which means you obtain any fungi or bacteria, but we work with fungi from either leaves or, in our case, the fungus is underneath the bark. So this is called canker. So you have to peel the bark to find this pathogen. And the pathogen itself creates these they look like bruises basically, but they're smaller cankers, and you have to culture them in a non-specific media, which means non-specific media really means that there is no specific culture to grow your pathogen in. That means everything else that's on that particular branch will grow there as well. So that makes the isolation of our pathogen a little bit more complicated. So what we found in the eastern U.S. is a lot of pathogen, which is actually used as a biocontrol. It's called trichoderma, and it's associated usually with native trees, and oftentimes it's just incorporated into soil as an amendment, and it creates better, healthier trees, and it prevents other pathogens of attacking that particular tree. But when we isolated, that's what we found in Tennessee, and when we did the same thing in California, where the trees are heavily infested, we found almost none of the trichoderma. So this could be one of the reasons that we're seeing differences in the East versus West, because we're finding a lot of basically native fungus associated with trees, which is one of the lines of defenses the trees have. But we're talking about native trees in this area. So now we're taking that basically natural product that's associated with the tree, which is also commercially available as a biocontrol. So we are using that to inoculate some of the trees in the greenhouse and look for the gene expression, basically to see what genes are turned on and off when we introduce that biocontrol and when we don't. So we're trying a number of different things to see if we can save the trees.
That's interesting about the some of the regulatory hurdles that you face and some of the logic behind it. I think it's kind of there's the lab leak hypothesis for COVID is a hot topic right now. So I think some listeners will maybe kind of hear some similarities there, which in my opinion, whether it is or isn't a lab leak thing, to me it feels a little bit like availability heuristic sort of thing, where it's kind of easy to imagine a movie of like, these scientists were meddling with Mother Nature, and then this crazy thing happened and mutated and got out of control. Where usually it's just usually like someone brought firewood to the wrong area, and that's what happens. It's a little more clumsy and nuanced and subtle than that. I mean, I think that's going to be one of the hurdles, which is. I think a lot of people get their ideas of science from TV, and TV jazzes things up quite a bit, and it's not super nuanced. And I love complexity and nuance. And I think it's endlessly fascinating to get lost in and keep diving into the fractal-like complexity of all things. But But a lot of people kind of want to be hit over the head with things a little bit, and it's a little more exciting to imagine mad scientists in labs and that sort of and you know, the more easily you can picture something, the more readily you believe that it could be the case, availability heuristic, and
So speaking of TV,
Yeah.
can I give you
Sure.
two things that always
Hit
drive
me.
me crazy in CSI based
Oh,
dramas?
CSI. Oh man, they're so
Just
brutal. Yeah,
Now, I'm just going to
hit me.
focus on two. Number one, since we're dispelling things, PCR does not take 5 minutes. So, when they isolate DNA and then they need to confirm the match of their suspect, and they
Yeah.
put it in a fancy machine, and then they get results in 5 minutes, they don't. Number one. Number two, these really expensive shows have really bad pipettes. They all have pipettes from the early 90s. I was like, please invest in just a nice set of pipettes. You can get it for $1,500.
Wait, what
It's
are they? But
pipettes? They're used basically for dispensing or obtaining very small amounts of liquid. So usually under 1 ml, and we use that for
Okay.
DNA isolation.
Okay.
And they're always old pipettes.
That's the thing that gets you. That's funny. I just I like that there's I'm a big Christopher Nolan fan, and you can tell that he does his research a bit more. And so when they're talking about things, it's things where like the concepts make sense within the world that he's building, and he's not just throwing around fancy sounding words. He's maybe building fancy concepts. But a thing like CSI it's so because they just say words that they know the viewers aren't going to know the meaning of. So it's just like, ooh, a smart person is talking right now. And I think that going back to the idea of making science communication more is I also think that makes the idea of science more unnecessarily intimidating to the average public than it needs to be, because they see CSI. And first off, if you actually know science, that stuff's a joke. All the stuff that they wrote on the board, none of those numbers add up. None of what they're saying actually makes sense. But for the person that doesn't know that, I imagine it's really intimidating to hear. Whereas a lot of the most profound concepts in, say, evolutionary theory are really, you don't need fancy words to explain them. You can explain them in a way that anyone that's willing can wrap their head around. And certainly I would never want to make fun of someone that doesn't have the capacity to learn this stuff. But in most cases, that's simply not the case. Most cases, it has a lot to do with a willingness. And sure, there needs to just be better communication, generally speaking.
That is one of the things I really, really imprint on my students. Usually I try to explain to them writing words that you need to Google is not a good science communication, even in scientific journals. As a non-native speaker, you look for synonyms quite often, right? And sometimes you put them into places like, this does not make any sense, but somebody else needs to tell you that part. But I usually tell them the way you're going to give every presentation is, number one, use images as much as you can to communicate, because people do not want to read. If I talk about my disease, nobody cares the name of, again, unless you're really doing exactly what I do. But people do not necessarily want to remember the pathogen name. They won't remember the vector, but they will remember the story. They will remember the pictures, and the images will be, what's the expression? The pictures are worth 1,000 words. And also, if you can break down these complex issues, and again, even when we give presentation in our own department, we have such a diverse group of people. We have entomology and plant pathology department together. So our entomology students and faculty, they are not necessarily pathologists, and vice versa. I mean, I have basic knowledge and I took several classes, but that does not make me an expert by any shape or form. And any additional explanation with entomology area would be appreciated, and vice versa. So I usually tell them, you have to give an explanation and presentation like your mom will understand, who does not know anything about science or does not have any interest in your project. So you have to make her interested in your project. And they've been actually pretty good about it. So I had a student who's working on microbiome, and how do you explain microbiome? It's a pretty complex issue, and it's a fascinating topic. But if you can I had actually people after one of my students presented his seminar, and they said this was one of the best presentations I've ever seen. And he was like, this is the first time I completely understood what the microbiome studies are all about. And I was like, that is a good science communication in its core. So now you captured audience that you probably wouldn't usually, and then they're willing to ask certain questions. And this is, intelligent group of people. So it's not, you're talking to scientists. It's not even a non-scientist group. Right. So, and that is, I think, where we fail as scientists in general, and where we need to do a better job. And it goes back to even what I do today. I do everything but physical, basically more or less no lab work these days almost, or very limited lab work. But I do everything else that I haven't been trained for. Everything from budgeting, financing, managing people, managing projects. So all the things, and science communication, all the things that I have never had a single class. And so you're just basically learning as you go. Some of us are more successful than others, and you pick up tips from your colleagues, and you learn better ways, and you learn using your mistakes too. But I encourage my students, especially PhD students who are interested in continuing further, take some financing classes, take professional development classes. I think it's really important, and especially science communication, how to simplify the message. Because at the end of the day, if you cannot reach the audience, it doesn't really matter. You're not communicating well.
Yeah, one thing that I find myself thinking quite a bit about is just how much kind of top-down thinking there is out there amongst people that aren't educated in the sciences, and there's the amount of explanatory power and thinking about evolution and some of these bottom-up kind of emergent properties of all these different systems working together. It just it has so much more, I guess, like I said, explanatory power to it, and it's not intuitive. I'm sure we're the first species ever that's that's been able to kind of conceptualize these bottom up processes. I'm sure every dog or deer or what a mammal on the planet thinks there's probably a big deer in the sky taking care of all of us, or whatever else, and conceptualizing things that way. It's just kind of intuitively how life feels in the way that Earth feels flat if you aren't taught any differently. And rather than it's kind of an unfortunate thing is that maybe this is a little bit too much of a chip on my shoulder or something, but I just think that evolutionary thinking is such a powerful tool that really can help improve all of our understanding of this existence and improve life for us. And it seems like there's people view it as like an attack or something like that on their personal belief system or something, or people view evolution as like, oh, that's neat, cool, things evolve, okay, we stood upright, terrific, but they don't realize all the different ways in which these evolutionary factors and complexities play into every little thing in life. And now you have something like, say, Thousand Cankers, and because someone's moving wood around, and there's these different symbiotic and ecosystem relationships that now you're that's the other thing, is people don't quite understand how it benefits them usually unless it hits their pocketbook directly. So unless you can say, hey, you're a carpenter, your price of wood is going to go up because of this, which means the price of your houses is going to go up, which means it's going to be more cost prohibitive for people, or this poor person making the casket maker in Italy that makes the finest walnut caskets, and
Mm-hmm.
now he's having to charge more. He or she is having to charge. Women make walnut caskets too. Sorry for that little. And
You can't even die.
and yeah, now you've saved up your whole life. You're Italian. All you want to do is be buried in a walnut casket. This is what you've been living for. And just in that last year, the price bumped up just enough where it's like, I guess I'm going in pine. I guess all that work was for nothing. So yeah, I don't know. Just a little rant that I go on from time to time about.
So take home message: don't die yet.
Yeah.
Hold on.
So I mean, I know you work a lot with your with students and people that are interested in science. How do you think you know, other than maybe giving CSI a few tips on how to make something a little more scientific, how do you think we go about explaining some of these things to the general public? I think that if you're in an area where 1,000 cankers isn't maybe killing off your trees and you're not as attached to it, and you aren't noticing, you aren't in the market for a house or building a house and knowing that wood prices might be fluctuating or something like that, I think that it's harder for people to it doesn't, you know, people work 40, 50 hours a week and they're tired and they want to get done with at the end of the day and watch CSI. And so, I mean, it's just something that I constantly think about. How do you better reach people without like getting in their face like, no, listen to, even if you don't want to listen, I'm going to make you listen. I don't think that works at all. I mean, most of what I do is cater to a group of underserved people that are interested in this sort of stuff and just don't have a real outlet for it, and provide, because I kind of gave up on the idea of converting people.
And that's the thing. I think if we learn one thing during this pandemic, nobody's getting converted to any way of thinking.
Right.
But you can provide options. So as a land-grant university, University of Tennessee's land-grant, so we have extension services. So I don't have extension appointment, but we can partner with people that do. We have a plant disease clinics, and which each state has one plant disease clinic. So people from all the constituents of the state of Tennessee, and every state in the United States has one disease clinic that's associated with a plant disease clinic that's associated with a land-grant university. So they can provide their samples. You may be a big farmer, or I can be just a random gardener. And you and I can both provide samples to the disease clinic, and part of their job is to serve the public and to provide you with recommendation of what is the problem, is it a disease or a pest issue, and how to control it, or what are the mitigation issues. So that can be one of the lines of defense. Also, for the state of Tennessee, for instance, we have extension agents in every county in the state of Tennessee, and other states have similar systems, either per county or per district. And those can be your partners. For people like me, I have a 90% research and 10% teaching appointment. So I don't have any outreach appointment, but I can partner with people like that. And I have done that in the past. There are a number of smaller conferences, number of field days or gathering where people want to learn different things. And you can present part of this in a less scientific, rigorous community where you can reach larger groups of people that would not necessarily come across your research or the things you do. And Forest Service is a really good outlet for us as well. And any kind of training with Forest Service personnel, and then they can trickle down that knowledge to their constituents. But I think where my passion is also, and I think a more meaningful and a long-term, not necessarily that the other one is less meaningful, but I think really where the real change happens is when you reach younger people, when you reach students at a younger age and get them interested in this. Not necessarily they're going to all do plant pathology or science in general, but I work in a number of programs that like there was a gadget girls, there was a girl culture, and a play on words for agriculture. But it's basically introducing these women these are usually high school students, middle school or high school students, and showing them that science can be fun. So I'll use two examples that we did, and it's not even in my field. We worked with I do believe Zika. We didn't work with real Zika. I'm just going to say that. So what we did, we had a color change. So it was a
Here you go, girls. We have some Zika virus for you to play with.
So, and by the way, we could not use the word sexually transmitted. So we had to use a different word. I can't remember what the word was because these were minor. It's pretty fascinating. That's another thing.
Yeah.
But we used basically tubes that they would change the color when they get in contact with a infected individual, and the infected individual had a slightly changed color. So what we did is we grouped them like, okay, now you two are gonna basically you're gonna be infected by this particular I think it was mosquito, that particular situation. You're gonna get affected by this mosquito, and this mosquito is gonna affect 3 more people, and then we're gonna also have close encounters, or whatever the word we used, with 3 more people. So just to see how things spread when you exchange bodily fluids.
Mm-hmm.
And
can't say sex. We're just going to exchange our fluids.
I know, and I'm just going to roll my eyes on that one. But It was a very impactful experience. Again, they didn't do anything super high tech. They didn't do anything even in my field. But that got him really interested in, I was like, oh, this can be fun. This is pretty cool. So tell me more about it.
Mm-hmm.
And this is the first step in the right direction of educating people. And then they're going to start asking questions, and they're not going to feel inferior because they do not know what this means. So you explain why we do certain things, and then you bring them to camps. I'm part of a really interesting summer project. They bring high school students. They're from juniors to, I think, freshmen. They come to the university, and this is the first time for them to be at a university setting. They work in different labs, and they work in my lab with grad students. And they just learn random things. So I had a girl that came one year. They come in pairs, and one year they're intimidated by everything, which I completely understand. And I asked them to we were cleaning some tissues so we can plate them and put them in clean culture so they can see what pathogens are on these leaves. It was a very simple project, nothing super fancy, because they're with us only for 8 weeks. And then the idea is they're going to do the presentation of that project and PowerPoint and poster, and they're going to talk about that. They don't know anything about plant pathology. They don't know anything about the things we do. They don't even know what fungi are for the most part. So she had a total meltdown because she said, I'm really bad at math, which every single girl almost says when they join the lab. And she was like, I cannot calculate 10% bleach solution. And I was like, no worries. And, it's like, I couldn't do that at some point either. So let's do this together. And I asked everybody in the lab who was there at that particular moment. I was like, let's do calculations. And I was like, I'll show you that everybody's going to do it slightly differently because everybody's mind functions slightly differently. We're going to get to the same, because I do math slightly differently than the rest of my lab. And it happened that every single person really did it slightly differently. And I was like, first, this shows you diversity of a thought. I was like, every single person here did the math in a slightly different way. And number 2, we're here to help you. So to disseminate that fear of, I'm working with grad students, they're super smart and I'm not. And then at the end of the day, at the end of 8 weeks, actually, I have to show you what she did because it was pretty fascinating. So it came from somebody who actually had a meltdown of not knowing how to make 10% bleach solution to, at her presentation, she did this. She was like, I know everything about fungi. That's how she started her presentation. And I was like, nice. This is awesome. It's 8 weeks. Yeah. It doesn't matter that she's not necessarily going into plant pathology, but I got one girl excited about science. And somebody who was very intimidated to make a 10% bleach solution to somebody who was knowing everything about fungi. And I was like, good for you. I don't even know everything about fungi.
Yeah.
But that excitement translates into so much more. And that was only one summer, one student. So my mission in life is to have these students every year of my career, from high school students to undergrads in my lab, that get this training, that learn new things just because it's there, and then their mind gets completely blown away. That things we do, the complexity of fungal world, and also the classes I teach, I really try to incorporate enthusiasm of my research, not to bore them to tears by any shape or form, but to tell them like how big this world is. And as a result, each year I have several students that are either interested in grad school, or they're interested in undergraduate research opportunities, or they're interested in getting internships just based on that class. And I call that a win. So, and that is my little version of science communication that I can do on my small scale. I may not change the world, but I can make a difference in a few students' lives, and that's what I can take.
Very cool. Have you ever heard of the Asian female math study? They had. I'll just tell you. They had. They had female Asians. They had in the control group. They had them take a math test, and then in just regular, they had regular scores. In the experimental groups, they had them. Before they took the study, they asked them like their name and then their race, and so they're primed to remember that they're Asian, and then they performed better on the math study. And then on the other one, they had them say their gender, and they're primed to say female, and they did worse on the math test than in the control. So there's a lot of self-fulfilling prophecy and kind of psychological stuff that happens with that. There's speaking of getting more females in science, I remember you having a pretty interesting background. Before we leave, do you want to share kind of how you got into science in the first place?
Sure. So originally from Bosnia. I don't know if I said that or not. I was born and raised there, and I came to the U.S. When I was second semester sophomore. But I also grew up in Bosnia during the war. I was 13 when the war started. And I'm just going to use the "what doesn't kill you make you stronger" phrase. So it was interesting upbringing. I'm originally from Sarajevo, so we lived under the siege for 4 years. And you just get completely different appreciation for life, for important things. And you learn not to sweat big things, because at the end of the day, you don't know if you're going to be here or not. So when things calm down, which means really when you realize this is going to last for a while, things are not going in any other direction than you're under the siege, and this is going to be much longer than what we anticipated. We had to resume life to whatever the normalcy was there. So my high school was in downtown, and I lived in suburbs. But what it really meant, the city does not have electricity, and the city does not have water. But I also have to cross 15 to 20 sniper places in each direction to get to my school.
Wow.
And it's usually an hour and a half in each direction. So going to school was literally a gamble, and you never knew if you were coming back home alive or not. So that gives a completely different meaning to life. And you just learn to appreciate all the wonderful things that happen to you. And so fast forward a few years post that, I survived. I survived all those sniper places. You learn to run really quickly. And you just find humor in things. And that's actually how we really, at the end of the day, survived. It is having each other's back. And I had a group of students that live in the same suburb, and we would always go to school together and hoping for the best. And so the opportunity presented itself for me to come to the U.S. To continue my education, and I got some scholarships, and I had amazing. I call them now my American family. They're really not officially my family, but they've been integral part of my life, and they eventually helped me. And I had amazing people who supported me along the way, and few advisors that got me interested in research. And I went and presented that research at a national conference, which was the way I met my future advisor, who at the airport actually offered me assistantship to get master's degree with him. And I was like, you haven't even seen my resume. You haven't seen my GRE scores. But he said, the fact that you presented as undergrad at a national conference tells me enough about your drive. And I was really lucky enough to have people like him and my undergrad advisors in my career for the last 20 years. And I still have an amazing mentors, which is one of the ways I really want to give back and provide the same that was provided for me. They saw something in me. They saw potential. They saw person that I couldn't see. And now I see this through my students, which is really a beautiful circle of life in a way. When they doubt themselves, where they don't think why I give them certain opportunities. I was like, if you could only see your brilliance and the beauty of your mind the way I see it, I would love for you to have my eyes for a moment and just see how amazing things in life you're going to accomplish. And I was like, this is probably how my advisors believed in me. And this is why I'm so passionate about, in a way, giving back, because this was given to me. And sometimes students just need that little push, or a person who's going to believe in them. Or I'm on another mission of providing these. Recently I started recruiting a few veterans, actually, which was another passion, because that's the world, even though I haven't been a veteran myself, but that's the world that I came from. Mm-hmm. And we have much more in common than we don't. And providing opportunities for these students and showing them what's out there, and just basically giving them opportunities to learn in the lab, and just telling them all these fellowships, scholarship opportunities that they were not aware of at all, is really amazing. And not just for them, but for all of the students in my lab. Because I paid my undergrad with scholarships entirely. I was not allowed to have a loan as an international student. And I told them, if I could do it in a foreign language, you don't have an excuse. So you can do it too. And I think there are so many ways, but students didn't know about it. And I learned about certain opportunities myself. I got a Fulbright a few years ago. As a citizen, I can do that now, but I didn't know certain opportunities like that existed. And I'm really promoting that among our students now. And also a lot of state jobs that are internationally based. Department of, not Defense, Department of Education, I think. They have all these amazing opportunities for students. I didn't know they existed. And I was like, if you guys need to do this in between, either after undergrad or during the summers, or something to open their minds in a different way. So I'm trying to provide these opportunities for students that are not aware of that. And I think that's part of my mission in life beyond science.
Very cool. Well, awesome. Well, I appreciate you so much, and thanks for sharing that. And
anytime.
Yeah, and thanks for coming on the show, Denita Hadziabdic Guerry, and yeah, it's a lovely. Thanks for accommodating me and coming back on to clear up some of the internet connection issues that we've had. But yeah, you are absolutely terrific, and I appreciate you.
No problem. Thank you.
And thank you, listeners, for being such wonderful, curious people. We'll talk with you next week.